afternoon, everybody. I have been working this project mainly investigated how brassicas cover crops reduce aphanomyces with disease. I'll talk about the importance of pea, problems and limitations of pea cultivation, and devastating disease aphanomyces pea root rot, disease control with brassicas cover crops. First, root and shoot tissue of brassicas cover crops, effects of root and shoot tissue on pea root rot, the combined effects, and then split it up root effects on aphanomyces pea root rot disease and the key organisms involved in ecosystem services, nitrogen fixing and nitrogen organisms community. And then finally the shoot tissue on inoculum potential of aphanomyces disease and pea root rot disease. So pea is very important crop. It contains high protein level. It's an excellent source for animal feed and also human consumption. Growing pea can fix atmospheric nitrogen by build from pea root nodule, and also use leguminous pea as a break crops in cropping sequence after a cereal to break down the pathogen networks. But production of pea is very difficult due to soil compaction. If the soil is highly compacted, pea plants cannot grow well. And also the devastating disease aphanomyces pea root rot, the causal agent aphanomyces disease. This pathogen can survive in soil more than 10 years without host plant due to have a very thick and protective cell wall wasteful. And currently there are no available resistance, aphanomyces resistance pea cultivars. And chemical fumigants are forbidden in many countries, therefore it's extremely difficult to grow peas for these limitations. And now we'll talk about the life cycle. When pea plants grow, it's root release flavonoids as a root exudates, used as an attractant to attract the pathogen inoculums. So when OS4, this OS4 gets signal that there is some pea plants growing, it's directly germinate by growing hypha and move towards the pea root. It's moved towards the growing pea roots, and some growing hyphae can produce sporangiums. Sporangiums is like a short mycelium hypha. It contains more than 100 zoospore. When zoospore hatch, can swim in water and move towards the pea roots. After encysted, the zoospore immediately germinate by growing hypha, and hypha can grow intercellular space of the pea roots and produce male antheridium and female ogonium. Antheridium ogonium. When the male antheridium Plasmodium fertilized begonium produce new spores. And finally, the necrosis disease develop until epicotyl region, then the plants completely collapsed. So in my studies mainly investigate this spore and this hyphal growth effects on Brassicas cover crops. The Brassicas mainly used for oil production, and in agroecosystems also used for as a cover crops to protect the soil from erosion and nutrient leaching in fallow seasons. So, however, the renewed interest to use Brassicas to protect the soil from disease pressure, because Brassicas tissue contain glucosinolates, and there is the distribution of a cell tissue, the glucosinolate and myrosinase intense distribution, and this figure showing how after growth incorporated in the Brassica shoot tissue in root and shoot tissue in soil. So there are three main different types of glucosinolates. Aliphatic is a linear structure, chemical structure. Aromatic is a cyclic, and indolyle is a semi-cyclic structure of glucosinolates. When these glucosinolates hydrolyzed by the myrosinase enzymes after mechanically disrupted the cell tissue, the water in plant tissue or equilibrated water can hydrolyze this myrosinase enzyme, glucosinolate, and produce volatile isothiocyanates, thiocyanates, and water solubles, nitriles, and epithionitriles. So when these hydrolyzed products interact with the pathogen, the pathogen reduce the pathogen ability to infect the host plant, or pathogen cannot produce spores after interact with these toxic chemicals. Current studies mainly, and the first paper is a review paper, mainly investigate mainly deal with the factors affecting aphanomyces, pyruvate disease severity, like different factors, so soil moisture, temperature, types of soil, and different strategy of disease control and their mechanisms. And the paper 2 mainly deals with the root and shoot effects separately on the development of aphanomyces, pyruvate. Paper 3 is mainly the root effects on the key organisms, as I mentioned earlier, the nitrogen fixing and nitrifying organisms community. And final paper 4 is mainly the concentration of isothiocyanates and the exposure, pathogen exposure types, mainly investigated on the inoculum potential of aphanomyces and the reduction of pyruvate disease severity. So I start with these pilot studies of several Brassicas species, Lapis alba, white mustard, Brassica juncea, it's Indian mustard, Raphanus sativus, oilseed radish. These three are Brassicas species belongs, and none these cover crops are grown in infested soil, artificially infested soil, 9 weeks until flowering stage. When it reaches at flowering stage, the cover crop's root and shoot tissue cut, chopped, and mixed with the infested soil, and peas were grown in infested soil for 5 weeks to observe the development of disease severity in a greenhouse biosphere. So from these experiments, the results showing this Brassica reduced Mendocinophis alba significantly reduced disease severity compared to non-Brassica, and also from if you contrast control techniques. But from these experiments, it's not very clear these effects from Brassica is after incorporation or during this 9 weeks growth. So in the following studies, they split it up root and shoot effects. And another objective of these studies was to select some cover crops in following studies. So these studies is mainly the root growth effect on phonomyces and observe the pre-root growth disease severity and the abundance of nitrogen fixing and nitrifying organism communities. So here these cover crops are grown in 11 weeks until reach at flowering stage in a different nutrients level, low and high. And before harvest, these cover crops, soil sample collect for volatiles and soil samples were collected. These volatiles are collected from root rhizosphere soil from here to investigate analyze the toxic isocyanates can produced during this growth 11 weeks, and also analyze the DNA for the key organisms, determine the abundance of the key organisms of ammonium oxidizing archaea, ammonium oxidizing bacteria, and that's all involved in nitrifying and also nitrogen fixing communities. After 9 weeks, root and shoot removed from the soil and sieve the soils to remove the major portion of root tissue and leave it for 2 days to allow to remove the volatiles if some volatiles left in the soils. So, and then peas are grown in that soil for 4 weeks and observe the disease severity in a greenhouse biosphere. And soil samples again collected from that soil after 4 weeks and compare this the abundance of key organisms, nitrogen fixing and nitrifying organisms, have any relation with the development of disease severity. So results showing the Brassica cover crops in the Mendocinophis alba when growing high nutrients level significantly reduced disease severity compared to control. It's more than 5-fold decline the disease severity. So this is the control, how looks the control oriented disease severity, and this is the how reduce the disease severity. think this Brassica synaptis alba is when we analyze the collected volatiles, we found this synaptis alba produce 3 times more aliphatic isocyanates compared to Brassica dulcea. So that this is reduced from hair to hair, mainly I think is the more aliphatic isocyanate released from synaptis alba. That is the more possible reason to reduce disease severity compared to Brassica dulcea. The interesting thing is here, we didn't find any suppression from the abundance of ammonium oxidizing archaea. The gene copies in a per gram dry soil is 10 logarithm transformed in here. And none of any of the organisms ammonium oxidizing archaea, ammonium oxidizing bacteria, nitrogen fixing organisms. So in ammonium oxidizing archaea, in the case, does added nutrients significantly enhance the abundance of ammonium oxidizing archaea compared to velocity. And the other treatments did not differ significantly. So in this studies mainly investigate the volatiles from the shoot tissue, as I mentioned earlier in the outlines. So shoot tissue from Brassica shoot tissue, and from fresh condition shoot tissue volatiles effects on the inoculum hyphal growth, and volatiles from dry shoot tissue. So the objective was if we transform the fresh to dry tissue, air dried, does it work? Does it contain the glucosinolate and produce the volatile substance? So that's why we have split it in dry condition and fresh conditions. So it is something like a this volatile from fresh tissue and the hyphal growth is something like a put a plastic cup and added tissue on the cup and add some water and immediately placed their fungal pure culture, pure culture in the petri dish on the top of the cup and seal with the laboratory flames and leave it in a growth chamber for 3 days and observe the hyphal growth, how hyphal growth diameter increasing every day. So this is the this in vitro experiment mainly investigated the volatiles, how interact mycelium, hyphal growth of pathogen, and this greenhouse experiments mainly this the fresh shoot tissue incorporated in infested soil and immediately sound pea roots, peas in incorporated soil and observe the pea root plot disease severity during 4 weeks greenhouse biopsy. On the other hand, the volatiles from the shoot tissue in these experiments it's a big difference. So first I put a sieve, it's a perforated sieve, and placed a filter paper and add mycelium, waste spore of Aphanomyces dry spores, talcum powder mixed in here, placed on the top, 4 gram, and closed, something like this. I just kept it in like this, and another cup, this I showed it earlier, placed the dry tissue, a certain amount of dry tissue, and attached this system, upper system on the top, and worked with the laboratory films and leave it in a chamber for 10 days to allow to interact the produced volatiles with the waste spores. After 10 days, remove the waste spores and mix with the fresh soil and use for greenhouse fibers for 3 weeks. So that is the and here is this in vitro experiments. In this I used only single doses, and here is used increasing doses of dry shoot tissue. The objective was to look at which volatiles from which doses can completely prevent the fungal growth in a 3 days growth period. And this time exposure is from the highest dose from this experiment. Further investigated, does it require 3 days to expose the pathogen? So in this experiment, time is only the highest dose, which got lethal effects, split it in a different times, like a 40 minutes exposures and 80 minutes and 120 minute exposures, to look at which exposure time is enough to completely inhibit these pathogens. So that is the in-depth investigation. And also collect the volatiles. This is the collection procedures. And during the time, according to this experiment, during the different time spots. And finally, analyze the glucosinolates profile in a dry shoot tissue. So let's start with the results. This in vitro experiment showed this the leaf tissue of Brassica juncea strongly affect the pathogen growth. Also the stem of Brassica juncea. But when this tissue incorporated in a soil and observe the pre-root development, it doesn't no reduction. On the other hand, the nutrient statements when introduced to the piece on the same day, significantly reduced pre-root disease severity. And I think that these nutrients contain some salts which can affect the pathogens. That is my idea. So in here, when expose the spore, and after 10 weeks exposures, the spore from Brassica juncea reduced disease severity significantly, nearly 4-fold different disease severity. And this figure showing during the 3 weeks how developed the plant's growth compared to Scalopsigelos, Synaphis alpha, and controls. So that means the So this dose experiments showing this, if the dose is increasing, the pathogen growth is decreasing like this. And I found the highest dose completely prevent fungal growth and no further growth if you placed it in a fresh condition after 3 days exposures. So this study is 3 days and I measured the hyphal growth. So then I thought that this happens also the synapse alba same, but the magnitude is very low. But from these studies, I thought the 3 days growth is enough to suppress this pathogen, or it's need a couple of hours maybe. Then I took this highest dose of only the Brassica juncea and further investigated in a different time, exposure times, like 40 minutes, 80 minutes, and 120 minutes. And then I found this 120 minutes is enough. There's no need to keep 3 days to completely prevent the pathogen and no further growth after 2 hours of exposures at 24 degrees centigrade temperatures. So glucosinolates profile to Brassica dry shoot tissue showed this synapse alba contain 2 times more amount of glucosinolates than Brassica juncea, and synapse alba dominant tissue dominant by aromatic cinnamon glucosinolates. After hydrolysis, this particular parent's glucosinolate produced aromatic benzyl isothiocyanates. On the other hand, Brassica juncea are dominated by aliphatic cinetine glucosinolates, and after hydrolysis, it produced allyl isothiocyanates. So here is the interesting results. I have collected the volatiles during 2 hours of hydrolysis in different time points in 30 minutes, from 0 minutes to 30 minutes, 30 minutes to 60 minutes, and 120 minutes, just as a exposure time, and have found these the volatiles from allyl isothiocyanates from increasing over times when hydrolysis during the 2 hours. On the other hand, benzyl isothiocyanates started with high peak and immediately is get down. So these are the 2 dominant isothiocyanates in 2 different plantations. But the common aromatic isothiocyanates, phenylethyl, that's the followed the trend according to the dominant isothiocyanate releasing trend in both cases. But both are the same chemicals. This produced the same glucosinolate called gluconestrate. So the ratio of transformation from gluconestrate in a Brassica juncea to produce phenylethyl isothiocyanate, the ratio is 11.5 conversion ratio, and on the other hand, in synapse alba 15.5. So this difference, releasing difference, I think, is the how dry shoot tissue dry shoot tissue, how quick absorb the water when hydration towards hydration process going on. That is their characteristics, different characteristics of these 2 tissues. So finally, conclusions. So aliphatic isothiocyanates from Brassica synapse alba mainly reduce disease affect the economics and reduce electrodigestibility, but did not affect nitrogen fixing or nitrifying organism. This is the process like a low doses but longer exposures in root growth. In aliphatic isocyanates from Brassica juncea shoot tissue strongly inhibit economyces utis in a closed system, but did not work if incorporated in a soil because the volatiles evaporated so quickly and left the pathogen unaffected. And finally, the effective isocyanate concentration and exposure, these three factors are very important if you think the potential biofumigation concept to apply in agricultural systems. So these are the three